Let's get started. I was having some technical problems. So the plan for today is we're going to keep talking sleep mechanisms. We should be able to get into circadian rhythms as well. And then Monday, we will keep talking about this, finish off anything we have for the lecture, and then the sleep homework stuff is due at that time as well on Monday. So we'll talk homework on Chapter 19 on Monday. Also, paper summary number 2 is due at midnight. And then I've been promising this quiz forever, I swear. I've gotten 6 questions written. I thought I'd be able to get it done before class today, and it didn't get done. So I'm sitting there trying to write question 7 and watching a blinking cursor. But it will show up soon. And you will have an adequate number of days. It's not going to be due at the end of the weekend at this point because I didn't get it to you. So you'll have a decent number of days to get it done. Anybody have any questions about anything? Okay. Well, so if you remember what we were talking about, actually I'm going to step back where I was. So we were talking about different explanations for sleep. So we talked about adaptive explanations. So from, what is the selective advantage to sleeping during the nighttime? And for us, there's quite a bit because we're not very good at moving around and using sensory input at night. There's restorative arguments about why we sleep in terms of maybe replenishment of glucose supplies within brain, which has kind of limited support. But I think much better support are these ideas about restoration in terms of removal of waste products from brain. And this is something that apparently does happen, particularly during really deep sleeps. So I've got yet another TED Talk. I like this one quite a bit also that talks this out. So I feel like there's sort of 2 halves to his talk. One of them, I think, links back to something we did back in Chapter 7. You remember this neuroanatomy worksheet? And at one point, I asked you to draw the path of cerebral spinal fluid as it's moving through the brain through the ventricle system. And we did, right? And so you probably drew something like this on your sheet. The idea is that we make, this isn't showing the lateral ventricles up here, but we make cerebral spinal fluid starting up in the lateral ventricles of the cerebrum, and it goes down to the 3rd ventricles, through the cerebral aqueduct to the 4th ventricle, and then some of it goes down the spinal canal, some other parts of it emerge and then circulate around the entire brain. His argument is that this is an incomplete story, though, because some of what happens is that once we get outside the brain to the surface of the brain, then we would have, I don't know if I can do this real easily. Nope. All right. I'm just not going to worry about it. But then we would have, during sleep time at least, we would have passage of cerebral spinal fluid from the cortex, from the outer surface of the brain, back down through. And so it's sort of channeling through and then whisking away waste products as it goes. So it's not just a completely one-directional thing. There's, the cycle really would be sort of completed where you have a cerebral spinal fluid passing back down through the brain from its surface and then ultimately, again, merging through this way. And then, some component of it ultimately is delivered into the veins. But there's a little bit more of a cycle, he would argue. So I think that was cool. The other thing that I think is really interesting is this argument about what kinds of waste products are actually emerging from this system. And so the one he really honed in on is amyloid beta. So I remember I heard this story first. I was just listening to TED Talk. I listen to podcasts on my drive because I don't have one radio station that stays in tune. So I just do all podcasts during my commute. I literally had to pull over when I heard about amyloid beta being one of the waste products, because it just completely blew my mind. So I pulled his paper, and so here is a paper that so this is out of Jeff Iliff's lab. Z is the lead author, but Jeff is one of the final authors, which is sort of like place of importance. But in any case, this is from his lab, and this is the experiment that he referenced where they're trying to monitor beta amyloid. So here, what they're showing here is over the course of a time period, this is the level of beta amyloid recovery. So this is basically how much beta amyloid is cleared out once injected. I think it's the way you would interpret it. And there's 3 different treatments. There's an awake treatment, so they're doing this and monitoring us while the mouse is awake. This is during sleep. And then this is ketamine. So ketamine's an anesthetic, and I think they're kind of using it as a positive control for sleep. You don't have to worry about the ketamine line, really. But if you look at this, what this is showing is that when the individual was asleep, they very rapidly basically recovered all of their beta amyloid. So they're sort of starting off, I guess, I don't know, I don't like the way I'm phrasing it, but they're basically starting off with large amounts of beta amyloid present when they sample at each of these time periods. So they're sort of starting at, I don't know, what is it, about 15 minutes, a half an hour, 60 minutes, 120 minutes, and so on. So they are initially recovering most of the beta amyloid after they injected it, but then as they start to wait longer and longer time periods, if you're asleep, there's less and less beta amyloid that's present within the brain. That's why this line drops down so quickly. Whereas the awake brain doesn't clear that beta amyloid nearly as quickly. So much more of it is still present after an hour than was present in a sleeping brain. This is a little bit easier, so they've kind of converted these rates into a rate constant. So basically a larger line here indicates faster rates of clearing of beta amyloid. And so what you can see here is that there is a significant difference, that's what this bar indicates, between the sleeping brain versus the awake brain, where the sleeping brain is clearing beta amyloid much more quickly than an awake brain. So again, not proving beta amyloid or that like lack of sleep causes Alzheimer's. That's several significant steps down the road in terms of this research, but it is indicating that you're getting better clearance rate of beta amyloid. And beta amyloid, as we saw all the way back in chapter 2, when we watched a little video on Alzheimer's disease, that is one of the hallmarks, is that we get accumulation of beta amyloid oligomers that form these plaques. That's something that clearly does happen during Alzheimer's. And we see beta amyloid being important in other conditions as well. Chronic traumatic encephalopathy, you'll sometimes see it there as well. So there's reason to think that this is a really important finding. And I think we'll see more of that over the coming years. So that gives us a feel, again, for sort of why we sleep. So, again, I'm sort of referring you to as far as memory consolidation and improvement, that was sort of Russell Foster's video. We talked about adaptive explanations. And then we're also talking here with Jeff Phillips' work about some of the restorative functions of sleep. So clearly sleep seems to serve a really important role. And the last thing I'll point out is that when we're calling what we're calling sleep in this case, this is non-REM sleep. This is just like classic deep sleep. Okay, and that seems to be something that's happening as neurons are doing these big up and down oscillations. Okay, so if that's why do we sleep, then I guess the next question is how do we sleep? So what is the mechanism that underlies sleeping? And it's a complicated story. In order to really understand it, I think one of the things you have to understand is that we have what are referred to as these diffuse modulatory neurotransmitter systems. And there's a whole chapter earlier on that has a section on these. So I'm going to kind of refer you back to that chapter, although I thought I did so by number. I think it's chapter 15. If you need to find these figures are from that chapter. But the idea is that that not all neurotransmission that happens within the brain is just like really obviously where you have like one cell talking to one other cell, and there's sort of like this continuous series of neurons getting signals. We have these sort of large neurotransmitter distribution arrays that send neurotransmitter all over the brain. And at certain times a day, like when we're awake, some of these will be more active, and when we are asleep, these large diffuse neurotransmitter systems will tend to be less active. So for example, this one's describing a norepinephrine system. So there's this area of the midbrain called the locus coeruleus, and it's transmitting norepinephrine. And so like here's the center of that nucleus. The cell bodies would exist here, but they send axons all over the brain. So they send axons into the cerebellum and down into the medulla and further down into the spinal cord and all the way up into the cerebral cortex through this sort of arcing system. And all this way there are axons delivering norepinephrine all over the place. And it's sort of generally resulting in certain kinds of excitation to certain kinds of neurons. Similarly, we have a serotonin system. So serotonin is generated down here in the brainstem. These are these RAF nuclei that we talked about in pain modulation, but they are also responsible for producing serotonin, and they send that serotonin all over the brain. And so when you are active during the day, you're bathing neurons in this serotonin neurotransmitter. Similarly, there's an acetylcholine system. So here's a center that's generating acetylcholine and sending that all over the brain. So again, the same kind of an idea here where we have these large diffuse systems where there's axons distributing a neurotransmitter all over the place and resulting in sort of generally excitatory signals that are active during wakefulness. So when we are awake, what tends to happen is these systems, so norepinephrine, serotonin, acetylcholine, are distributing these neurotransmitters all over the place. And they will tend to cause neurons all over our brain, therefore, that have receptors for these neurotransmitters to be more active than they otherwise would be. So that you're increasing alertness, increasing wakefulness as a result of these systems. As you start to get tired, these start to shut down. So for example, one thing we talked about that tends to trigger sleepiness is adenosine. Adenosine builds up over the course of the day. That's why we take caffeine, because it helps to inhibit the receptors for adenosine, but eventually it'll overwhelm the caffeine. And not everybody does caffeine, that's just sort of my problem. But as you increase your adenosine concentration, adenosine will inhibit the release of these neurotransmitters and thereby sort of decrease excitation across the brain. And again, that's going to result in a decrease in wakefulness, and therefore you will feel more tired. You also have other systems, so histamine, hypocretin. These are also released, so either by the midbrain or the hypothalamus. These things are all sort of released and also are amongst these sort of diffuse modulatory neurotransmitter systems. I should have indicated that I think these are generally excitatory as well. I really want you to focus primarily on norepinephrine, serotonin, and acetylcholine. Although I will point out hypocretin shows up in your homework because this one is particularly involved in narcolepsy, so that makes it kind of an interesting neurotransmitter. But the idea is, again, big picture, when you're awake, these neurotransmitters cause general excitation and they inhibit any kind of rhythmic firing that would cause sort of large-scale synchrony that results in those big delta waves that happen during sleep time. Whereas adenosine acts against these neurotransmitters. If they're not causing excitation, then you're going to have more rhythmic firing and you'll start to enter into more of a sleep state. So these guys are sort of anti-sleep systems. Last point I'll make is that at least some of the cholinergic neurons that are related to acetylcholine promoting wakefulness, some of these are also involved in shifting you from a deep sleep to a REM sleep. So as you shift during the night, you're still asleep, but as you shift into a REM sleep, you'll see some of these cholinergic neurons turn on and increase your acetylcholine production. And that is apparently at least part of the cause for what's going on that's causing parts of your brain to become more active during those REM sleep patterns and result in the beta waves and stuff that we see during REM sleep. Here's a picture to try to understand in terms of those brain rhythms. This picture is trying to depict these things. So we mentioned most of these neurotransmitters on the prior slide. So here's norepinephrine. So in the presence of norepinephrine or acetylcholine or serotonin, which has this annoying abbreviation 5-HT, but that's serotonin, or histamine, what ends up happening is you have, some of your rhythmic centers of your brain tend to get shut down. So what they're recording in this measure is actually the recording from the thalamus. And so what you can see in here, these kind of look like action potentials, but they're not actually really an action potential. They're a little bit longer waves. So what they're doing, going from this picture to this blue one down here, is they're sort of blowing this up and expanding the time axis. And so if you record from what the thalamus is doing, you can see it sort of is getting excited and then fires several action potentials in a row. Hopefully you can see that, but there's like a bunch of action potentials in a row. And then it kind of goes silent again. And then it comes up and fires a bunch in a row and then goes silent again. So this is the thalamus kind of doing its rhythmic firing pattern. And this is what's going to be happening when you're asleep. So this is a recording from the thalamus, but the thalamus is broadcasting this signal all over your cortex, all over other parts of your brain, and it drives the rest of your brain to sort of behave in this kind of rhythmic fashion where you don't have good coordinated action between individual neurons. It's everybody's kind of following suit. So this is non-REM deep sleep, but what happens is when we wake up, we start firing our excitatory diffuse neurotransmitter systems like acetylcholine and norepinephrine and serotonin and histamine. And the result of this is that all of these guys is that the thalamus shifts from this kind of rhythmic firing pattern to a much more active kind of firing pattern, where it's not, there's not sort of big up and down undulations. Now we've got sort of high activity going on in the thalamus. And there's more specific information being transmitted over the course of this time. If you think about what the thalamus does, so the thalamus, we said, was the gateway to the cerebral cortex. So any kind of sensory information that's coming in from your sensory systems, be it vision or touch or sound, whatever it happens to be, those systems get routed to the thalamus, and then the thalamus, in turn, relays that information up to the cerebral cortex. So one of the things that's happening here, and it kind of mentions this now, is that when you are asleep, you have information coming into the thalamus from your sensory systems. But the thalamus is kind of like ignoring that, and it's just kind of doing these up and down undulations. So it's not really paying attention to the sensory information that's coming in, which means, therefore, it's not going to relay that sensory information up to your brain while you're asleep. This is why you're basically disconnected from your sensory systems while you're asleep. You're not awake anymore. And you're not conscious anymore of that sensory information because it never gets to your cortex. It basically gets to the thalamus and just doesn't do that doesn't cause the thalamus to respond, because the thalamus is sort of locked into this rhythmic firing pattern. But once these diffuse modulatory neurotransmitter systems start firing and they cause the thalamus to exit that rhythmic firing pattern, now that's what you're seeing here is individual cells within the thalamus are firing, and they're sending then signals that convey information about sensory input that the thalamus is receiving up to your cerebral cortex. So that information is encoded up here. And now you're conscious and aware of your sensory systems. Does that make sense, more or less? Yeah. That's a good question. I don't know. So yeah, so the question is, does this have anything to do with being in a coma? And I don't know. It would make a lot of sense that it would be, but I don't know the answer to it. I have to look it up. I might try to go do that, because it certainly would explain why a person that's in a coma doesn't seem to be aware of sensory input. Certainly not doing much motor output at that time either, but if this would disengage their ability to get sensory input up to their cortex when you have the thalamus kind of in this pattern. Other questions? Try to remember to look that up. Here's another look at what's going on in the brain. So when you're sleeping versus not sleeping, or also when you're in deep sleep versus REM sleep. So the prior slide's kind of showing the thalamus is like this central pattern generator that's determining a rhythmic pattern of firing for much of the rest of the brain. And it does result in pretty dramatic differences between when somebody is awake versus when they are asleep. What we're trying to do in this picture is that these are not images that are showing one particular brain at one particular spot. They're more of a composite image. So what they're trying to do is, in these MRI setups, they are monitoring, I guess they have to be fMRIs. They are monitoring how much activity is going on in brains at one state, and then they're comparing it to the activity that's going on in a different state. And so the colors that are depicted up here are comparative. So if we look up here, this is REM sleep versus non-REM sleep. So in all these cases, a person is sleeping, but they're showing that when we're in REM sleep, we're seeing, and these are sort of, I think these are actually looking up from the bottom is the way they tend, that's sort of the convention. So you're sort of looking from the spinal cord up. But this is basically indicating in the red and the yellow areas, these are areas where there is more excitation during REM sleep. And then in these purple areas, this is indicating that there's actually less excitation, less activity, brain activity going on when you're asleep. And they're measuring activity basically by how much blood is going to the different areas. So an area of the brain that's more active tends to get better blood supply, and you can detect the blood supply using these MRI systems such that you can get a feeling, or not a feeling, you can get an indication of what parts of the brain are more active. So when you look up here, if you're comparing REM sleep versus non-REM sleep, I think what we tend to see is that some of these posterior parietal areas, so this is like the posterior back end of the head. This would be the front of the head up here, so these would be side to side. And what you're seeing here is that some of these areas in the posterior parietal cortex tend to be really, really active. And there's a lot of these sort of this central area that tends to be really active during REM sleep compared to non-REM sleep. And this sort of central area is limbic system. Do you guys remember what these things do? What posterior parietal cortex or limbic system, like what are some of their functions and roles in general? Like we definitely talked about posterior parietal cortex, right? But limbic system, you might do. Yeah, so limbic system is like this loop of brain cortex or brain areas, not cortex. They tend to be fairly deep, that are involved in emotional responses and regulation. So that seems to be upregulated when you're in REM sleep. What about posterior parietal cortex? Remember what that's doing? And it's parietal cortex. So parietal cortex is all about sensory information, right? What's the posterior parietal cortex doing? Remember? So it's definitely getting some touch information, yeah. Yeah, so it's getting somatosensory. It also gets, so the idea with the somatosensory system is that we came into like the anterior parietal lobe, areas 3A and 3B, and then we relay back to areas 1 and 2. But then we get into areas 5 and 7. And so areas 5 and especially 7 are more the area that's lighting up over here. So like, you know, those sort of initial somatosensory areas that you arrive in, those would be over here where there's actually not a lot of activity. But the area that's lighting up in REM sleep tends to be this area of the posterior parietal cortex where we have more like the higher order processing of sensory information tends to be coming in. So that does include somatosensory, touch and proprioception and that kind of thing. But it's also an area where those kinds of signals seem to intermingle with cells that are processing visual signals or auditory signals. So we sometimes call these multimodal association areas. And I tend to think of these as areas where we probably have some of our conscious perception of sensory information. So you see like this area is really lighting up as being pretty active where we do kind of our conscious perception of sensory information, even though like the somatosensory cortex per se is kind of silent. It's pretty blue or purple in this area, so it's not getting a lot of input. And this area over here, this is occipital lobe, so that's like visual input. And so the visual cortex is kind of silent too, but these visual association areas that are intermingling with the somatosensory areas, they're pretty active. And so we've got this system, this situation where you have like a high amount of emotional response areas running, and some of your like conscious perception of sensory information areas are running, even though like, you know, you're not actually seeing anything at this time because your eyes are closed. You're not maybe really feeling that much because those areas are down. So we kind of have a lot of sensory kind of, I tend to think of this like the conceptualization I have of this is that we've got a lot of sensory processing and consciousness going on. And we also have a lot of emotional things going on. And those things to me, that kind of sounds a little bit like a dream. Where, you know, they tend to have a lot of emotional responses in dreams. You are sort of aware or have this feeling that there is sensory information that you're perceiving, even though it's not real, right? So we may see things effectively in our dreams, even though our actual visual cortex that's processing real visual information is quiet. But this area that's involved in sort of downstream regulation of vision, it's running. And so I kind of think that's indicative of what goes on in terms of dreaming when we're in REM sleep. I don't have beautiful data that argue that's exactly what's going on, but it makes sense anyway. Here's a comparison of being awake versus in REM sleep. So I think this is kind of an interesting comparison because REM sleep, it looks so much when you're looking at an electroencephalogram, it looks an awful lot like being awake when you're in REM sleep. Lots of beta waves and such. So here they're trying to get at sort of the next level of information. So what areas are really excited? What areas are not very excited when you are awake versus when you're asleep? So I guess I should probably rephrase these because what the graph is showing, or what the figure is showing, if they're red, you are, let me make sure I get this right. Yep. If you are in red or yellow, that's indicating you're getting more activity during REM sleep than when you're awake. Whereas if you're blue, like this whole area up here, you're getting less activity in REM sleep versus when you're awake. So you can kind of stare at these for a second. So again, you can see there's like not much coming in from your visual input, but there is still a lot of sensory processing that's going on in REM sleep, even more so in some areas. Like this is all posterior parietal lobe. So there's even more sensory processing going on here when you're in REM sleep versus when you're awake. But your whole frontal lobe, like there's nothing going on. Your frontal lobe's kind of shut down, and this is where, you know, this is where your personality and like rational thought and that kind of thing isn't really running that well. And so that might also help explain a little bit about what's going on when you're in REM sleep, and that you have these sort of bizarro dreams that don't make sense, and you make bad decisions and other things that happen because your whole frontal lobe seems to be kind of conking out. But again, your sensory areas, sensory processing areas are really running strongly. If you think about, so again, here's, I guess I'm just going to read through this because it's kind of saying what I think we just said. So going from non-REM sleep to REM sleep, we tend to see this explosion of cortical activity, especially common in sensory association areas, but not in the primary sensory areas, like not in the visual area. That's what V1 is your primary visual cortex back here. It's also very active in the limbic system. If you're comparing REM versus being awake, you have not a lot of prefrontal cortex activity, so you have poor processing and poor thought, poor interpretation, sort of like rational interpretation. But you have lots of association cortex activity, especially in the posterior parietal cortex. So maybe you've got a lot of vivid sensory information, but it's not always comprehensible, and you're not interpreting it very well, perhaps. In terms of mechanisms and brain areas that are driving REM sleep, as you're going from deep sleep into REM sleep, as I kind of mentioned, you have a selection or a collection of cholinergic neurons that are present. They're coming out of the pons, but they start to fire, and they start to excite some of your brain cortical areas. So you're going again from like completely kind of shut down in deep sleep into REM sleep by turning on just the cholinergic neurons. At the same time, some of these you will have, at the same time that you're triggering your cholinergic neurons, you also have a bunch of your spinal neurons that tend to be inhibited, so you have paralysis. And this is where if you have a failure to inhibit these neurons, you can run into REM sleep behavior disorder, which again, as I've sort of mentioned, comes up on the homework in a sort of humorous way. This guy named Mike Birbiglia has a great little narrative. To stop REM sleep, what we do is we start to fire noradrenergic, so these are norepinephrine neurons, and serotonergic, so these are serotonin generating and releasing neurons. From the locus coeruleus and the rave nuclei, respectively, these start to fire and these turn off REM sleep. So sometimes acetylcholine generating neurons are referred to as REM on cells, whereas the REM off cells are the norepinephrine and serotonin ones. So here's a depiction of that over the course of, I think this is over the course of hours. I'm sort of losing my x-axis, but yeah, this is hours over the course of a sleep night. So here they are in deep sleep, deep sleep, deep sleep. So this is non-REM sleep. But then to trigger into a REM sleep, you'll have the acetylcholine neurons kicking on. These are the REM on cells. And then these guys eventually become inhibited as we start to fire the REM off cells, norepinephrine and serotonin. And then these cause you to shift out of REM sleep. And so then you return to deep sleep, and then again you'll go through another REM period. And so we have this sort of collection of neurons that seem to drive us in and out. I do think it's interesting, though, that these same neurotransmitters are also associated with wakefulness, right? So norepinephrine, serotonin, acetylcholine, if they're all sort of firing together, that's kind of what happens at the end over here, because this is when we tend to wake up. And these neurons, along with hypocretin and histamine and all these other ones, those neurotransmitters are causing us to ultimately be wakeful. But they seem to have this other kind of role, at least at sort of moderate release rates that causes to shift in and out of REM sleep. Okay, questions? So that's kind of like mechanism behind deep sleep, mechanism behind REM sleep, how we switch back and forth. Last little topic I'll just sort of throw in for this section before we get into circadian rhythms is about dreams. And in a way, I don't have a lot to say about dreams, because I don't know if anybody really should have a lot to say about dreams, because we just don't know that much. There's this long history of trying to interpret them and understand them. They are this really interesting phenomenon. If you've ever read Carl Sagan's Contact, dreams are identified as like this sort of unique human thing by the extraterrestrials in that book. Is sort of like this really cool thing that we can do. But there's certainly a lot of question about what do they mean, if they mean anything. So Sigmund Freud sort of thought of dreams as being really important, and so this is a way that you can identify wishes and fantasies, and so dreams sort of serve as a fantasy fulfillment mechanism. And so there's a whole line of work based on his hypotheses about this that I don't know are all that well supported at this point, but they do exist, and it is this line of work. There's another hypothesis, and there is some support for this, that the dreams actually might be directly associated somehow with some of the memory consolidation and enhancement stuff that Russell Foster was talking about. So an experiment, there are these Tetris experiments that I always found kind of interesting, where they would have participants in these experiments practice Tetris. Do you guys remember Tetris? It's like, I think it still exists, but it's, this game, you sort of turn blocks around and you try to build flat layers and then they go away. So they would have people practice Tetris prior to going to sleep. And one thing they definitely found is that if they had people practice pretty much immediately before going to sleep versus 4 or 5 hours before they go to sleep, people would show better improvements in Tetris performance by doing it right before sleep. So there really is something to be said for example, studying in the hours leading up to your sleep as opposed to leaving a huge gap, because it seems like something happens in your brain after you're able to go to sleep that sort of builds upon and enhances those memories and strengthens synaptic connections, and there's good support for that kind of thing. One thing they found is that also some of the people in one of these studies that I saw, they would report like dreams of like blocks coming down, and they're like trying to manipulate them in their dreams. So maybe there was some argument that maybe the dreams were somehow involved in memory enhancement. However, in some of these other studies, to test that idea, they somehow would try to deprive people of REM sleep. So basically, they'd watch you as you're sleeping. These are all these sleep center studies. So you're going to sleep with people basically monitoring you the whole time. They'd watch you until you entered a REM sleep, and then once you did, they'd like stop you and they'd wake you up. And so they compared people where they prevented them from going to REM sleep from other people that they prevented at different times of night, but they had the same number of wake-up events and that kind of thing to try to keep it controlled. And they found that depriving REM sleep did not seem to reduce the improvement in performance after sleep. So this is indicating that the benefit that you get to enhancing a task or practicing a task definitely has to do with sleep, but maybe the REM part of the sleep doesn't really matter, because if you deprive of REM sleep, it didn't seem to hurt their ability to consolidate memories, enhance skills, that kind of thing. Okay, so yes, sleep helps with memory enhancement, but maybe REM sleep, I don't know, the data I've seen have indicated that REM sleep doesn't have anything to do with that, which is too bad. That'd be cool. Another explanation, and this one is maybe the least satisfying to a lot of people, but this is from Hobson and McCarley, is something they call the activation synthesis hypothesis. And it's almost kind of arguing that dreams are accidental mishaps of how our brains are wired. So the idea here is that many of the associations and the memories that we sort of perceive during dream states are activated by some random discharges that happen from the pons when you're in the midst of REM sleep. And so the pons then, by sort of randomly discharging, is stimulating certain areas of our cortex, posterior parietal cortex, limbic system, these kinds of things. And our cortex then is, so the cortex then is receiving these apparent sensory inputs and limbic system inputs and trying to synthesize these into something that makes some sense. But they maybe are just sort of driven by these random firings of the pons and therefore might not have any real function at all and just could be kind of accidental byproducts, which I don't know. honestly, I'm kind of attracted to this idea. At the same time, I guess it's kind of sad if it's true, because maybe it means that they don't mean a whole lot. Although then again, you could argue that for them to be activated, even if it's from random firings of the pawns, you have to have those things in there to begin with, so maybe they do mean something about what's going on in your brain. I don't know. So, you know, I think that the answer to this and the summary of this whole slide is kind of like a, you know, I'm not sure anybody really has a good feeling for what's going on with dreams, why we dream, and I would probably argue that anybody that says otherwise is probably not right. There's lots of hypotheses out there, not a lot of data that strongly support any of them that I've seen. All right, that's a pretty good stopping point. So what we'll do on Monday is we'll pick up with circadian rhythms and we'll talk homework. So I will see you guys then. I will say there's a question on there that asks you, it's going to kind of push you a little bit to try to understand a genetic gene expression mechanism that explains circadian rhythms. Do your best. We'll try to explain it in class 2. We'll see how you do.